This lecture will cover the opportunistic mycoses in Chapter 59, pages 793 through 798. The opportunistic mycoses are saprophytic, which means they live off dead and decaying matter, and so this can sometimes make it very difficult to diagnose them since they can pretty much be found anywhere in the environment, and sometimes it's difficult to know when it's grown on a culture plate if it's an actual contaminant. So you really got to kind of judge where it is on the culture plate as if it were, you're looking to see if it were actually growing within the culture streak where you put the culture onto the agar, or whether it's growing outside of the streak. So those are things and skills that you will learn once you're in the lab to be able to recognize when something is a contaminant. So these organisms typically cause some sort of tissue invasion in individuals who are immunocompromised. So it's very important to make sure that you're working up this fungus when it's an immunocompromised patient. They're typically acquired through various aspects throughout the environment. So it could be situations where there's construction or demolition, especially in older buildings, any sort of remodeling, and sometimes there can even be situations where it's acquired through a hospital visit, that you get an infection, a hospital-acquired infection through these organisms. Aspergillus is found throughout the environment. The conidia can break off very easily, so you can see up here in this image where the conidia are just kind of attached to little phyllodes. So those conidia can break off and can go throughout the environment through the air and are breathed in to the host. It typically affects the immunocompromised host starting with a pulmonary infection, then it can progress to more of a disseminated infection. Aspergillus is seen in the clinical setting a variety of ways. Three of the ways that we typically see them are through allergic reactions, or through the production of a toxin, or through some sort of opportunistic type infection. Aspergillus causes a wide variety of disseminated infections. Some examples would be a fungus ball. It could be of the sinus or it could be of the ear, so you can see that example here. It could be cause an allergic type reaction. It could affect the sinuses. It can affect the heart or central nervous system. An Aspergillus infection can be detected using an antigen protein-based assay where you look for beta-D-glucan in serum, although there are other organisms that can possess beta-D-glucan. It is a rapid growth organism. It usually matures in about 3 days, but there are some species of Aspergillus that are a little bit slower with their growth rate. When we look at the macroscopic morphology, it can tend to aid in identifying the species of Aspergillus based on the color of the colony. Aspergillus fumigatus is a thermotolerant organism, so it's able to withstand higher temperatures, so temperatures up to about 45 degrees Celsius. So here you have a color chart of some of the more common species of Aspergillus and what the surface of the agar looks like with the colony, and what the reverse side of the agar looks like with the colony growing. So for the microscopic identification, this organism has a septate hyphae. So you can see in these images where there is a division in the hyphae at the bottom. And you also have an unbranched conidiophore. So this stem does not branch off, it just comes straight up. And it's attached with a foot cell. So the stem itself, or the conidiophore, comes down and out, so it attaches onto the septate hyphae. There's no division where it attaches to the hyphae. It actually has sort of like a little foot. So that's called a foot cell. Then the vesicle itself, which is this little radial type head up here at the top, it is attached either through with the attachment of phthalates that are attached directly to it. So you can see this image over here where you've got the phthalates that are attached directly to it. So you can see where they're like little finger projections coming off. Or they can be attached to a metula or metula, depending on where you're at, how it's pronounced. And so you can see those are a little bit larger, and then you have the phthalates attached to them right here on the end. So I'm kind of marking that in there. The phthalates have conidia coming off of them, and the conidia form chains. So you can see all of those little long chains coming off of both of these species. So here's a list of 4 of the most common or more common to what you'll see in the lab. They all look very similar as far as the Aspergillus species, with how the phthalates are attached to the vesicle and given this sort of dandelion look. But what you're looking for to distinguish one from the other is the vesicle itself, how it looks, as well as the phthalates and the conidia, how they're attached. So with Aspergillus fumigatus, you'll have a very large vesicle. So you can see a large vesicle here. And then what you'll have is bottle-shaped type of phthalates that are covering the surface. And so you see how they're just kind of, it's really hard to see really that what they are, that they look like a bottled shape. But they're coming up straight from the vesicle. And then from there you have all of these little conidia that are attached to them, and they're just kind of hanging out there. Some of them have already come off, so you can see those around on the side. So they're really small. With Aspergillus flavirus, you've got a very large vesicle as well. It's kind of going to be big. And again, those phthalates and conidia will be attached to that vesicle, except the main difference you're going to see is they may have a little bit of a tint to them, a yellowish orange tint. With Aspergillus niger, it is a spherical type vesicle. So it's going to be rounded all the way around. And you can see an image of that here at the bottom. It's kind of hard to see that vesicle, but it is rounded. And with those phthalates and those conidia, you're going to have them coming all the way around it. So you can see this one here that's laying on the side. So you can see this conidia come up, and then you've got that rounded vesicle in there. It's like a little spear shape. But then you have all of these phthalates with the conidia attached to it coming all the way around it. And you can also see that it's a very dark brown color. So that is very characteristic of that species. And then with the Pterias, you've got, it's going to be more of a part of a circle, or a half of a circle. And it's going to be, the surface will be covered as well. And it produces a very large type conidia. And that's it. And so here are just some more images of some of those where you can see how some of them are shaped a little bit different. So you can kind of see in comparison of these two where you've got this shape versus this shape of the vesicle. And then they just kind of come up, attached to the metula or the phthalates, or the metula and phthalates, and then all of those come up. You can see how long these chains are right here. And then again, some of the more classic images, but they all still have that similar look. You can see that little bit of a yellow pigment in these right here in the center. So you can see that little yellow pigment that's coming off of those. And of course you can see the dark pigment, but they all look very similar with that little dandelion look. Fusarium species cause a variety of infections like sinusitis, wound infections, particularly in burns, allergic fungal sinusitis, or an actual infection inside of the eyeball itself. With the macroscopic appearance, it is a rapid growing organism. It is fluffy to cottony. It can be a variety of colors like pink, purple, yellow, green, and you can see in the top image where this organism is of shades of pink, a little bit of white, and also has a little bit of yellow to it as well. So it looks similar to cotton candy, but this is probably not one that you want to eat. When you look at the microscopic appearance, there are small septate hyphae. So here at the bottom, you can see some of those small hyphae, and I believe that may be a septa right there. The hyphae give rise to phyllids, and I don't see any good images of the phyllids attaching these macroconidia. But the macroconidia are shaped similar to boats with a little sickle type look. So they have like a slight curve to them. And you can see those pointed ends like a sickle would be. And then you also have those septate divisions inside of that macroconidia. So that makes them multi-celled. So there's multiple divisions with multiple cells inside of it. Geotrichum conidium is an opportunistic organism, so it will cause an infection in the immunocompromised host, usually through ingestion or inhalation. For the macroscopic appearance, these colonies tend to be white to cream with a yeast-like look to them, but they can also be wet, dry, powdery, similar to a ground glass appearance. So if you look here at this image, especially right here at the periphery, you can see how fine, the fine look that it has to it, but you can also see how it's kind of got this wrinkled yeast-like appearance to it as well. For the microscopic appearance, it has a septate hyphae, and what it does is it forms these rectangular cylinder-looking barrel-shaped arthroconidia. And what these do is they kind of form out in little chains, so they don't bud off. Even though some of these kind of look a little rounded, they don't really bud off. They just keep those little arthroconidia barrel-shaped types looks to them. So there's no blastoconidia, there's no budding that's happening with these. Acremonium species cause several types of infections. So infections of the nails, the eye, the heart, central nervous system, the lining of the abdominal cavity, and of bone. It grows rapidly. With an immature colony, the colony itself will appear yeast-like, but as it matures, it will get more of that mold-like appearance, and it can be white to gray. And you can see an image here in the center with that mold-like look. For the microscopic appearance, the hyphae are small and they are septate. They have unbranched tube-like phyllids. So these things that are coming off are actually phyllids. So they're not conidiophores. And you can see how it kind of forms like a little tube. But at the end of that tube, you have this little flower-looking structure here, which is all the conidia that are contained within a little cluster. And here at the bottom, you can see where that's kind of broke open. And so you can see those, how they have like a little bit of an elliptical type shape to them. The Penicillium species are very virulent for the AIDS patients, and they cause a variety of infections, like infections of the eye, inside the eye, the ear, esophagus, lungs, heart, abdominal lining, and urinary tract. For the macroscopic appearance, it has a variety of colors that it will present as, from various shades of green, and you can see an organism here that has various shades of green within that one organism. And what you're looking for on the microscopic view of this organism is you're looking for septate hyphae, and you're looking for something that appears very similar to a paintbrush, or it could appear, in some people's opinion, it looks like a skeleton hand. And so you're looking for that organism to, it may branch or it may not branch out towards the end of it. So you can see some branching here, or this particular one doesn't branch. But you see where the mitella is attached to phyllodes on the end. In this organism, the phyllodes attach directly to the conidiophore. And then on the end of that, you're looking for all of these conidia to make that paintbrush type appearance. So this one over here, you can see that those chains are very long, and this one gives more of that little skeleton hand type look, as well as this one. And then you can see all those conidia at the end. Mycoses causes a variety of infections as well, and I think you can probably kind of start seeing that these opportunistic organisms cause a lot of the same infections. For the macroscopic appearance, it has a rapid growth as well, and it can be velvety tan to olive brown, and it can also be somewhat powdery. So you see two examples here where you've got that very velvety type look, and then you've also got that look that's very powdery, almost kind of dry. For the microscopic appearance, it does kind of look similar to the penicillium as far as that look with those phyllodes and the conidia attached, except the difference with this is these phyllodes are really swollen. So you can see how they're kind of swollen at the end, at the base, and then they kind of taper off. So you get that, it has a different look than the phyllodes of the penicillium. And again, you've got the conidia that are in the long chains. Porcilium is a organism that appears like a lilac to various shades of lavender or pink, and you can see that here in this image at the bottom where it's kind of got that pinkish purple shade to it. The microscopic appearance, the phyllodes will be present in clusters, and they also will have that swollen base with the tapered neck appearance, just like the last organism. So you can see those where they're kind of swollen at the bottom and then they taper up. With these though, the difference between this one and the last organism is that these conidia, you can see where they're a little bit more elongated out versus that round conidia. Scopulariopsis is associated with a variety of infections within an immunocompromised host. For the macroscopic appearance, it's moderately rapid growth, and it is white initially, but then comes a light shade of brown, and it's very powdery. So you can see an example here in the top image. For the microscopic appearance, it has septate hyphae. The hyphae will have, off of it, it will have these hyphae-like conidiophores. So you can see those kind of coming up. Some of them look a little bit shorter than the others, but they're coming up. And then they come and turn into these lemon-shaped conidia that form in chains. So you can see some of those kind of have like a more of a rounded shape to them. So these are more rounded. And then you have this one over here that it's got more of a little lemon type shape to them. But these are a lot larger than what we have been looking at.